Swayam Prabha. Digital India. Educated India.
Hello, my dear students. Welcome to Swayam Prabha, sponsored by MHRD, New Delhi. Course title Medicinal Chemistry. Lecture title Measurement of Drug Effects. Faculty name Dhamak Vikrant Murlidhar. Module number 5. Lecture number 8. So let's see the contents of this lecture, measurement of drug effects. So drug effects, basically they are being measured by dose response relationship. So that will be the first content of this lecture. So basically there are two types of an dose response relationship. So one is graded dose response and second one is quantile dose response. After that we will look sketched plot. After that double reciprocal plot, and after that we will be looking drug receptor interaction forces. Now we'll see the concept of dose response relationship. So dose response relationship describes the magnitude of response of an organism as a function of exposure to drug after a certain exposure time. Dose response relationship can be described by dose response curve. So here you can see a dose response curve. So response, it is nothing but an biological activity, and dose. So this dose, it is taken into a log concentration. So bracket, these are indicating an concentration of a drug. So drug, or we can say dose. So dose, it is a different thing little bit, because dose is in such a quantity which shows an biological effect, and that quantity it is of a drug. So minimum amount, or we can say an amount of drug which is sufficient to make an biological activity is said to be and dose. So this dose response relationship, it describes the magnitude. So magnitude, it is nothing but a measurement of a response when it is been exposed to a drug, and this happens after a certain time or after an exposure to a drug. So here you can see that at minimum concentration of a drug, so there is no response, and later on it shows an sigmoid shape of a curve. So sigmoid shape of a curve, it is indicating that now from here the dose, it has been now an effective, or drug, it has been an effective, and after that we will get an sigmoid shape of a type of a curve. So all the time it happens in case of a dose response relationship, that it is not a direct relationship, a proportional relationship exactly with all the concentration of a drug, but it is always a sigmoid shape of a type of an curve, because when drug it comes to bind to a receptor, so it finds a binding site, and when there is sufficient amount of an drug present at an binding site, then only it will start its effect. So at very minimum concentration it will not give an effect, and after that it will rise, and after some time it will become an plateau. So the reason of this plateau, it is sigmoid shaped curve, and reason for this plateau, it is that the binding site has now no capacity to bind a new ligands or new drug molecules, and the binding site, or we can say a receptor site, so there is no site for a drug to bind, and that is why it becomes an plateau now, and so it becomes an S-shaped curve. All the times you will find such kind of a dose relationship. So what are the basic terminologies in dose response relationship? So we'll look over that. So dose of a drug is a specific amount or weight of medication. So dose of drug, so dose, it means that a specific amount of a medication. Medication, it refers to a dosage form actually. So for our language we will use a drug, specific amount of a drug or weight of a drug, like say 500 milligram of an paracetamol, it is a dose of an that particular paracetamol, because to show an biological response 500 milligram of an paracetamol it has been required in an adults, and dosage form in such a thing that it forms an unit dose. So unit dose, it means that dosage forms are been designed in such way. Suppose say paracetamol tablet, like example of an crocin, it is a brand name, and paracetamol, it is a drug in it having some excipient, and it is said to be in dosage form. So the dosage form, it is nothing but a tablet, and that tablet has a 500 milligram of an paracetamol, which is a dose of an that paracetamol. After taking an 500 milligram of an paracetamol, then only it will show an biological activity. At low doses, the activity will be minimum, as we have seen in the graph also. After that, EC50. So EC50, or we can say effective dose or effective concentration. So both terms are similar, effective dose as well as an effective concentration. And effective concentration, EC50 is the concentration of a drug that gives half maximal response. So from graph it can be understood. So it is an sigmoidal shape of an curve, and this sigmoidal shape of an curve, so here it is giving an 100% response. So it is up to an 100% response, and it reaches to an plateau, and after reaching towards an plateau at 32 log scale of dose, it shows an plateau region. But there are some errors to calculate such type of an doses. So it has been made in between that we can calculate an EC50. So EC50, it is, and we can say an effective concentration is this thing, that when it reaches to an 100%, so that is nothing but an effective concentration, or we can say an maximum response. So it is said to be sometimes Vmax. So that is nothing but an maximum response, and half of that it is said to be an EC50, that is effective concentration. So full concentration or effective concentration, it will give an maximum response, and EC50, that is half percentage. So here it is nothing but an 50% response, it is taken on 0.5 scale, and so it is said to be that 50% concentration gives 50% responses given at particular concentration. So that thing, it is said to be an EC50. After that, some terms like efficacy. So in earlier lectures also we have seen what is efficacy and potency. So again we will see it. So efficacy, maximum effect that a drug can produce regardless of a dose. So it can be understood by graph once again. And potency, amount of a drug that is needed to produce and give an effect. So from graph it can be understood that a response versus a log drug dose concentration. So here it is nothing but an drug A molecule showing an response, and after that drug B molecule it is showing a response. So the potency of an molecule of an A or A drug, it is more potent than compared to an B, because it is having a more biological affinity towards an receptor. So it binds fastly, but it binds slowly. So from this difference, it can be understood that, so at this concentration, or from a zero concentration or little bit uh, above towards an zero, so it shows an minimum is this shows a minimum affinity towards a receptor, and that is why, so it is after having a certain concentration only, so B will bind towards a receptor. At minimum concentration it will not bind towards a receptor, and when so this drug A, it is having more potency than drug B. And efficacy, it is little bit different thing, that see here it reaches towards an Vmax, but B, it is having more efficacy. So efficacy, it is nothing but an biological activity. So biological activity, or we can say a response, it increases for B, and so we can say that drug A is more potent, but drug B is less potent, but drug B is more having more efficacy than drug A. So drug A is potent and drug B is having more efficacy, or it is having more biological activity. And also of an EC50, see here EC50, as potency increases, so EC50 also decreases, right? So EC50 also decreases. So at minimum concentration, so it will show an potency. So for drug A, it is having an low EC50 value, but for drug B, EC50 value will increase. So EC50, it will come over here for drug B, and for A it will come over here. So plotting in perpendicular to it, so it can show EC50 value. So it is having a minimum EC50 value, and this it is having a maximum EC50 value. Now we'll see the first part from dose response relationship, that is nothing but a graded dose response. A graded dose response relationship describes a drug effect which increases in proportion to increasing drug dose. So in earlier graph also we have seen that as the concentration of drug increases, so the graph it is showing a sigmoid shape, and as the concentration of drug increases, so there will be increase in response also, and that is said to be a graded dose response relationship. A graded response to a drug is seen in an individual and increases with the dose. So it is also a disadvantage of this graded dose response, that it can be seen only an individual. Like if we like in case of an pharmacology, students are performing a practical on acetylcholine by taking some smooth muscle from an animal, and after taking smooth muscle like from an say ileum, basically ileum we are taking for an practical purpose. So as the concentration of drug increases, so there will be increase in the response of on that chymograph paper. So this thing, it is said to be a graded dose, and it can be calculated only into an individual. So that is also a limitation for a graded dose response. So it cannot be calculated on a population. Graded dose response graph plots a response to a drug against its concentration. So as we have seen in the earlier slide also, that dose response relationship. So graded dose response, they can be plotted with the response. So percentage response or biological response, we can say, against the concentration of drug. So we have seen with an log concentration of a drug, or it can be in some cases it can be in form of units like milligram per kg. So in such cases, or sometimes it can be in a log phase also. So there are some concepts regarding graded dose response. So maximum or Emax is the maximum effect achievable by that drug. So once a drug comes to bind towards a ligand, after binding of a ligand towards a receptor site, and once receptor site it is full of a drug, so it shows a maximum effect. And so that maximum effect also we have seen in case of a graph. So it is nothing but a maximum biological response. So here you can see a maximum effect. So it is nothing but 100%, and beyond this the graph cannot move. So beyond this graph cannot move, it means that so the sigmoid shape now it forms a plateau over here. So the meaning of plateau is that if we increase the concentration of drug even, so there will be no any change in the effect, or that there will be a plateau region over here. So it will be in the steep line. So it is indicating that now there is no binding site, and when it reaches towards this 100% concentration, 100% biological activity, so it means that now there is no active site present for an binding. So all the active site it is been saturated, and drug will show effect, maximum effect up to this only. The threshold dose or minimum dose. So name itself indicates that minimum dose, which is the lowest dose at which a drug effect is seen. So here you can see it is at lowest effect, lowest concentration of a drug, where we can see an threshold dose or minimum dose. So how much minimum dose does and any system requires or any tissue requires to give an biological activity? So that is nothing but an threshold dose or minimum dose. This may be represented by 0 point, but it doesn't have to be. Consider that before giving a drug, there may be some baseline pre-drug effect. For example, when the drug is noradrenaline and effect is arterial tone. So this is nothing but, and we can say this point it is nothing but an minimum dose. So where a drug molecule it shows an effect, but this effect it is at some concentration. So that is nothing but a minimum dose. So there is never a direct relationship. So always it will be a sigmoid shape type of a relationship in case of a dose response relationship, because when drug reaches towards a binding site, so it requires minimum concentration of a drug to bind towards a receptor. Then only it will bind, and after binding, it will exert its effect after some binding of some concentration. Now we'll see second concept from dose response relationship, that is quantal dose response relationship. Quantal dose response relationship describes a drug effect which is binary, either present or absent. Now we'll see next concept from dose response relationship, that is quantal dose response relationship. So quantal dose response relationship describes a drug effect which is binary, either present or absent. So the meaning of this thing, it is that binary. So to overcome a limitation of a graded dose response relationship, so in case of a graded dose response relationship, it is only for an individual candidate. But in case of an quantal dose response relationship, so it is binary. So it means that it is for an population, or it is for more than two individuals. So it may be either present or absent. So in some cases it will happen that some of the individuals they will show a response to a particular dose, but some of an individual they will not show a response to a dose. So it happens due to a different physiology of an humans, and also the threshold dose or minimum dose. So it also differs within population, and some general assumptions are been made when dose it has been calculated. So it has been calculated for a binary, so all the individuals in the population, they can have an effective dose for an effective biological effect. So dose it has been calculated on that basis. Once if dose it has been calculated for an individual, so it will not be suited for an another individual also. The quantal response to a drug is observed in population and is either present or absent in a single individual. So it will happen that it is observed in a population, so it is in large number, and in some individuals it may be absent or present, or it will not show any effect. Quantal dose response graphs plot rate of outcome occurrence in a population against the drug dose. So here you can see a standard deviation. So in the standard deviation, so this is a Gaussian plot or Gaussian curve we can say, and it is number of patient responding. So percentage of number of patient responding. So here you can see that at mu, a 4 milligram dose, it is been responded by 40 patients, or with a percentage of 40 patients. So this we can say it is an quantal dose response. But at lower concentration so the response it is different for different individual, as it has been said over here that it may be either present or it may be absent for a single individual. So the all individuals coming in this line, it will not happen that they will show a response at this 0.1 dose. So, and it has been also shown by this graph that at this particular dose, so all patients will not show a response, but at 4 there is maximum response. So 4 it has been calculated as a dose for any drug, and because a large number of population, it gives an biological activity at this position or at 4. So this it is taken as a dose of a drug, and as the concentration of drug increases, so there will be again loss, or we can say all patients will not respond accordingly. So some patient they will respond at 7 milligram per kg. Some patients little bit more, they will respond at 6. So maximum effect or maximum patient, they will show a response at this 4. So we can say that 4 is an standard one. Now we'll see next concept, that is scattered plot. So scattered equation is an equation used in molecular biology to calculate affinity and number of binding sites of a receptor for a ligand. Once a ligand it enters into an body, so it finds an receptor to bind it, and this scattered plot or scattered equation, it is used to calculate the number of binding sites and calculate an affinity for that drug. It is named after American chemist, that is George Scatcherd. So now we'll see an Scatcherd equation with the plot. So here you can see that R. So R, it is nothing representing an receptor, and plus L. So L, it is nothing but a ligand, or we can say a drug molecule. So R, it is available for ligand binding, and once R will bind to an L, means ligand will bind to a receptor, so it will form an RL complex. So now this concept, that is association constant, because when we come to a chemical reaction, always there is a dissociation constant or dissociation of an any molecule. But here it's an association constant, that receptor plus ligand, and now it has been associated. So association constant, it is denoted by concentration of an RL divided by concentration of R into concentration of an L. So it is different to an dissociation constant. So different to an dissociation constant means it is reverse to an dissociation constant, or it is inversely proportional to a dissociation constant. In case of a dissociation constant, so RL, it is at bottom, and R into L concentration, it is on the upper side. So it is opposite, association and dissociation. So these are the two reversible process, that a molecule will dissociate in this direction and it will associate in this direction. Now we require here over an association constant, and at an equilibrium at certain time, or we can say after a certain time, so there is remaining of an R and there is complex of an RL. So remaining of an R, it means that ligand, it is not ligands whichever were been available, so they have bound with a receptor and it has formed an RLL concentration, and this is nothing but an R concentration. So it is, and nothing but an receptors are available for an binding. So at equilibrium or after some time, it will show such kind of an relation. With a change in time, we can say T, it refers to an time. So after rearranging the equation, so we will get R equal to concentration of RT minus concentration of an RL. From the first equation, now replacing the values. So now replacing this R, concentration of an R. So now we will replace this RT, concentration of RT minus concentration of an RL in the bracket. So after rearranging the equation, we will get such kind of an equation. RL, concentration of an RL divided by concentration of an L equal to Ka multiplied by, in bracket, concentration of RT minus concentration of an RL. Receptor ligand and ligand concentration, and A, it is nothing but an association constant. So now we will plot and graph for this purpose. So here you can see on y-axis, it is nothing but concentration of an RL divided by concentration of an L, and concentration of an RL, it is on x-axis, and RT, it is nothing but an x-intercept, and K over here on a slope, that is minus K. So it is showing that as ligands bind to a receptor, the concentration, concentration of an R decreases as it forms an RLL complex. So negative sign, it is indicating that so there is no more availability of an R, because at certain level it will get saturated, and once it has been saturated, so there will be no binding for an ligand. So I will be explaining with an example how we. Now we'll look an scattered plot example, and from this, at the end of this slide, we will understand how from the scattered plot one can make an judgment or conclusion for a drug receptor interaction. So here you can see it is and receptors, and these receptors are having and ligands. So there are 4 receptors having 4 ligands binded with it. So from equation, right, Y, that is RL, and so the value for RL, that is, it has formed and 4 ligands has formed receptors with 4, the ligands have formed complex with a receptor. So the value it will be 4 because it is number in 4, and now there is no extra ligand present over here. So the value it will be 0, and the last value for Y it will be an infinity. And the value of X for an X axis, so it will be 4 again. So from here, from this it can be understood that starting from 4 and it will intercept at RL 4. Right? So we'll see some more examples so we can conclude it. So now here you can see that there are 4 receptors, 4 ligands have been joined to it, and 8 ligands they are remaining. So they don't have a binding site now, and these do not have a binding site, so these are remaining in biological fluid as it is. So now we'll come toward an equation. See, in the equation, Y equal to RL divided by L, concentration of an L. So RL, so these are number of 4 complexes, receptor and ligand, and total 8 ligands they are free now. Right? Concentration of an free ligands, that is 8. So the value it comes 0.5, and X, that is on the X axis RL. So RL it is nothing but an 4, as the value that is 4 for ligand receptor complex. So now if we come towards a graph, so from graph it can be understood that plotting on a 0.5 scale and stretching towards an 4, so this slope it is minimum compared to this slope. So from this it can be understood that for an effective, or we can say for an effective interaction, so number of ligands and number of binding sites, so they should be in the same concentration. But if we increase in concentration of an ligand, so the slope it will decrease. So from this it can be understood number of binding sites and number of an interaction or affinity towards an ligand. So for an affinity we'll take one more example. See, here you can see that ligands are free, but it is having low affinity. So low affinity it means that it is having a low potency, because they have formed a complex with an 2 ligands they have formed a complex with an receptor, but 2 are free. As 2 are free, but there are 4 ligands available, but they are not having an affinity towards this receptor, and so that is why they are free. Now we'll come towards an equation. So here in the equation RL, that is 2, so it is forming an 2 receptor complex divided by concentration of an L. So these are nothing but an 4 ligands, and after that RL, so RL complex of an receptor and ligand, so these are 2 ligands. So the value it will be plotted on a 0.5 on RL scale, and for RL, so it will be again minimum. So here slope has been decreased. See, from these 3 graphs, so it can be understood that so interaction, or we can say a slope, it is higher when concentration of an receptor and concentration of an number of binding sites, we can say number of binding sites and number of ligands are same, so we will have an higher slope values. But as the concentration of an ligand increases, so again it will the slope will decrease. But when there is low potency or when there is low binding at an receptor of an ligand, though the concentration of ligand it is higher, but once it is having a low binding property or low affinity or low potency, so the again slope value will decrease. Now we'll have one more example that it is also again showing, and that receptor are available, but these ligands are not having an affinity. So in the first example, there were 4 receptors with 4 ligands and in the fourth example there are 4 receptors with 4 ligands, but only one of them it is having a binding capacity towards a ligand. And now we'll come towards an equation. So in the equation you can see that RL, so this RL, so it is having one value, and L, concentration of an L, that is free ligands. So these are 3 in number, and RL again receptor and ligand, so that is 1. So value it becomes 0.3. So it is more minimum, or we can say less than 0.5, and we will get an slope very low. So from this all we can conclude that for an interaction towards an binding site, so concentration of ligand should be equal to an concentration of an receptor, but not even that. So it should have also an binding capacity or potency towards an receptors, so that can make a conformation into a receptor and it will show an biological activity. So depending on what desired action we require, so binding of an or affinity towards an receptor is also important. So this scattered plot explains that when slope increases, so it means that binding capacity of a receptor increases, and not even the concentration of a ligand by increasing just an concentration of a ligand is insufficient for an binding, but as the concentration of a ligand also increases, so there will be decrease in the slope value. So the meaning of this thing is that there should be sufficient ligands and sufficient receptors to have an binding, and that match should be perfect, then only the slope value it will be increasing, and from this it becomes an easy for a researcher to record an biological response or to note an biological response. And as there is low potency, so there will be less slope, and even if there is intermediate potency, still even there is minimum slope. Now we'll come towards an next equation, that is double reciprocal plot. So Lineweaver and Brook plot, it is also called as a double reciprocal plot, is a graphical representation of Lineweaver-Brook equation of enzyme kinetics described by Hans Lineweaver and Dean Brook in 1934. So this equation it is related with an enzyme kinetics, as we have seen in case of an receptors. So there are 4 types of an receptor, and one of an receptor it acts by an enzyme, or even G protein coupled receptor, that G proteins are responsible for activation of an enzymes. So for that purpose this double reciprocal plot it can be utilized. Actually this double reciprocal plot it has a significance that it can overcome an Michaelis-Menten equation. So we'll see one by one what happens actually. So significance of this double reciprocal plot is that, or Lineweaver-Brook plot is that, the most favored plot by researchers has 2 distinct advantages over Michaelis-Menten plot, in that it gives more accurate estimate of Vmax and more accurate information about inhibition. It increases precision by linearizing the data. So we have to calculate an Vmax value. So Vmax value, that is in earlier graph in dose response relationship, we have seen this Vmax value or maximum effect, and the half of concentration it can be calculated, and from that it gives a more accurate result. But there are some certain limitation with Michaelis-Menten plot, because Michaelis-Menten plot it is only on 2 axes, but double reciprocal plot, so it is on 3 to 4 axes, and that is having an advantage to calculate an Vmax value more accurately, and also by linearizing the data we will get an more precise values. See now coming towards an substrate, and this particularly it is related with an velocity. So velocity on y-axis and substrate. So as the concentration of an substrate increases, so there will be increase in velocity of an enzyme activity. So it is shown by a graph. that as the substrate concentration increases, the velocity of a reaction or velocity of an enzymatic activity, it increases. And as reaching towards a certain plateau, so it shows an Vmax. Now the problem with this plateau is that why it reaches an plateau, actually why it does not, it is not a direct relationship, because as the concentration of substrate increases, but there the affinity towards an enzyme, or we can say the enzymes, these are not available for now binding of an substrate. Right? So the capacity or number of we can say enzymes which are available at the binding site, so this has finished, and as this finished, so we will get an plateau, and we cannot calculate an accurate Vmax at that particular instant, because now there is a steeping value. Here it is a direct relationship, but after a certain time it forms an plateau. So this value, it is nothing but an infinity, because it does not increases as earlier it has increased with velocity and substrate. And as the concentration of substrate increases, so the velocity of a reaction or velocity of an enzyme kinetics also increases, but at certain level, so there we will get an plateau, and this, it is nothing but an we will say an infinity. And here you can see there are certain Vmax, and the values of this Vmax, so this changes with a particular at particular instant, and it is not now an accurate, it forms now an error, because if we consider this point, so it will not match with the second Vmax, and it will not match with the third Vmax, so it becomes an error in the graph. And there will be error in the Km. This Km, it is nothing but an Michaelis constant, so we will see in the equation. So as the Vmax, it shows an error, so there will be error in also in Km. And this is how in case of a Michaelis Menten equation, it happens that velocity against substrate concentration, so it has been plotted, and from that we have to calculate the value for Km. But this double reciprocal plot, it has an advantage on Michaelis Menten equation, so we can understand it by an equation itself. So for a double reciprocal plot, so we have taken certain values. So these values are, so V0, it is measured velocity. Vmax, it is a maximum velocity, and at this particular level, so earlier also we have seen the plateau region starts from here. So that is nothing but a maximum velocity at which it has been reached, and after that there is steep plateau. After that, S, it is nothing but a substrate concentration, and Km, it is Michaelis Menten constant. Now inverting an equation, now we have to, our goal is to draw an equation for double reciprocal plot. Now making changes, certain changes into this Michaelis Menten equation. So by inverting it, V0, now it becomes 1 by V0, and the values which are at denominator will become at a numerator, and vice versa. And after rearranging the equation, we will get such kind that Km divided by Vmax into substrate concentration plus concentration of substrate divided by Vmax into substrate concentration. So by rearranging, we will get such type of an equation. So now introducing a line equation for this double reciprocal plot. So we'll see one by one term. Y, it is for 1 by V0. After that M, it is for Km divided by Vmax. X equal to 1 by S concentration, and C equal to 1 by Vmax concentration. Now we have plotted a graph that is double reciprocal plot or line weaver brook plot, and now we can calculate a value for Km and Vmax accurately. So from the slope we will get a value of an Km and Vmax. So you can see it is now in linear one, and as we were having a problem with the plateau in case of an Michaelis-Menten equation, so it has been reduced or it has been minimized, and a limitation of that Michaelis-Menten equation calculating of an Vmax can be done with the help of double reciprocal plot. Now we'll see certain kinds of drug receptor interaction forces. So there are certain types of drug receptor interaction forces. So first one is covalent bonding. It is like we can also see such type of concepts in organic chemistry, because they are related with an organic chemistry in the field of medicinal. After that, ionic electrostatic interaction. After that, ion dipole interaction. After that, hydrogen bonding. After that, charge transfer interaction. After that, hydrophobic interaction. And after that, halogen bonding. After that, van der Waals interaction. So we'll see first one, that is covalent bonding. So covalent bonding is the strongest bond generally formed by sharing of an electrons between two atoms, and it is formed by a drug receptor interaction except with the enzymes and DNA. So this is one of the strongest bond that it is formed due to an sharing of an electrons. This interaction forms a strong link between drug and its receptor, but individually the interactions are reversible. So it can be reversed, that it is not an irreversible. So maybe it may be a strong interaction between drug and receptor, but these are also irreversible. Like example of an phenoxybenzamide at alpha hydrogenic receptor. So it is one of an drug, phenoxybenzamide, which attaches to an alpha hydrogenergic receptor by covalent binding. After that, ionic or electrostatic interaction. For protein receptors at physiological pH 7, basic groups such as amino acids, chains of arginine and lysines are protonated and therefore provide a cationic environment. Arginine and lysines are having in basic groups like ammonium or amino, and once it has been protonated, so it forms a cationic environment. So cationic, it is nothing but having a positive charge on nitrogen. Acidic groups such as carboxylic acid, again this arginine, amino acids, they basically contains two groups, that is carboxylic acid at one end, and at another end it is having an amino acid chain. And acids such as glutamic acid or aspartic acid are deprotonated to give anionic groups. So deprotonated means that carboxylic acid having H, it has been removed. Drug and receptor group will be mutually attracted provided they have opposite charges. So when there is interaction for a drug and receptor, this particular group, so it is forming it has been deprotonated, and this one is protonated, so it is forming a cationic environment, then it forms an anionic environment. So for a drug, it should have such type of an opposite charges group, so they will be attracted towards it. So and mutually this attracted group, they will form a drug receptor complex. Second, that is ion dipole and dipole-dipole interactions. As a result of greater electronegativity of an atom, such as oxygen, nitrogen, sulfur and halogens relative to that of carbon halogen bond in the drugs and receptor, where X is electronegative atom and have an asymmetric distribution of an electrons, this produces an electronic dipoles. So basically it means like an inductive effect, that is when it is having an electronegative atom attached to it and electropositive atom attached to it, so it will show an ion dipole moment. And these dipoles in the drug molecule can be attracted by ions or by other dipoles in the receptor, provided the charges of opposite sign and properly aligned. So drug which is interacting or which will interact with this receptors or receptors having and such type of an that carbon and oxygen, nitrogen, sulfur, halogen electronegative atoms attached to an carbon, so it will form a dipole, and this dipole they will attract a molecule having a dipole moment in or dipole interactions in it. So there will be dipole type of an interactions, and it should be that it should fit properly into an receptors. After that, hydrogen bonds. So also we have seen in case of an DNA alkylators, hydrogen type of an bonding. So hydrogen bonds are the type of dipole interaction formed between protons of an group of X. So X, it is halogen, and H, where X is electronegative atom or one or more electronegative atom. Y, containing a pair of non-bonded electrons, like in case of an carbonyl group or in case of an amino group or in case of an sulfhydryl group. So it forms an hydrogen bonds with an adjacent one. The most significant hydrogen bonds occur in a molecule where X and Y are nitrogen and oxygen. X removes an electron density from an hydrogen, so it has a partial positive charge which is strongly attracted towards a non-bonded electron Y. So whenever hydrogen bond it is formed, so X removes an electron density from an hydrogen, so it will as it is an electronegative, so it will attract more electron towards it and forms a partial charge. The interaction is denoted by a dotted line. So this interaction, it is nothing but an hydrogen bond interaction, and Y, it is nothing but a non-bonded electron like oxygen or sulfur, and this X, it withdraws an electron from an hydrogen, and as it withdraws an electron, so it is due to an inductive effect that as it withdraws an hydrogen, as it withdraws an electron from an hydrogen, so it will become an electron deficient, and this electron deficient is been fulfilled with some non-bonded electrons, that is like sulfur and nitrogen and oxygen. So this non-bonding electron, it will form a bond with an hydrogen, which is partial one. Charge transfer complex. When a molecule that is good electron donor come in contact with a molecule that is good electron acceptor, the donor may transfer some of its charge to an acceptor, and this forms charge transfer complex. So name itself indicates that charge it has been transferred, and it forms, after transferring of a charge, it forms a complex, and it requires a donor and acceptor for this purpose. Donor groups contain certain pi electrons such as alkynes and aromatic moieties with electron donating substituents on a group that contain a pair of non-bonded electrons such as oxygen, nitrogen and sulfur moieties. Acceptor group contains electron deficient pi orbitals such as alkynes and aromatic moieties having electron withdrawing substituents and weakly acidic protons. So the difference is that it is also having an pi electrons over here, but these pi electrons are electron deficient because it is having an electron withdrawing substituents like carboxylic acid, amino groups, not amino groups, carboxylic acid, it may be aldehyde groups. So all containing in C double bond or carbonyl are electron withdrawing substituents, and electron donating substituents such as OH or NH2, so or sulfhydryl. So these are nothing but an electron donating substituent present on this alkyne or aromatic moieties. There are groups on receptor that can act as an electron donor, such as aromatic ring of tyrosine or the carboxylate group of an aspartate. After that, halogen bonding, it has now been established that covalently bonded halogen atom can act as an electron acceptor. So it is nothing but an Lewis acid to undergo halogen bonding with electron rich donor such as oxygen, nitrogen, or sulfur. So these are Lewis bases, we can say, having such kind of an groups. These interaction can govern the conformation of an molecules in the binding site of proteins. So such kind of an interaction we have seen in the earlier lectures also, like in case of an cyclophosphamide, that is an alkylating agents. So it binds by such way. After that, van der Waal or London dispersion forces. Atoms in non-polar molecule may have temporary or non-symmetrical distribution of electron density, which results in generation of temporary dipole. So for creating an dipole, it requires an we can say a charge, or there should be an distribution of a charge according to electronegativity and electropositivity. But this case is different. It is in case of a non-polar molecules. Ionic molecules can easily form a distributed charge, that is plus positive center and negative center as it is. But when there is a temporary just generation of a dipole, as the atoms from a different molecules such as drug and receptor approaches to each other, the temporary dipole of one molecule induces opposite dipoles in the approaching molecule, and consequently intermolecular attraction is known as van der Waal forces results. Van der Waal forces are very weak forces, and these are only found in case of a non-polar molecules. These weak universal forces only become significant when there is close surface contact of an atom. So it means that a drug receptor, when they are very close to each other, then only such kind of an van der Waal or London dispersion forces can occur, and this occurs in a non-polar molecules. Now we will see a summary of this lecture. So we have seen dose response relationship, that is magnitude of response of a drug. So calculation of an measurement of an response. We have seen a graph with a sigmoid shape, that as the concentration of drug increases, the response also increases, but at certain level it becomes steep, or it becomes a plateau. After that, graded dose response relationship describes a drug effect which increases in proportion to increase drug dose. So we have seen with the graph that as a drug increases, concentration of drug increases, there will be increase in effect, and this effect or this relationship, it is said to be a graded dose response relationship, and it is for an individual one. After that, quantal dose response relationship. So this relationship, it is different from graded dose response relationship, as it is binary, or it is for an individual. It is not for an individual, it is for an population. Either it will be present or it will be absent, and it has been calculated for an population. And from population, we can calculate an accurate dose what has been required for a general population. After that, we have seen a scattered plot used to find the affinity and number of binding site of a receptor for a ligand. And also we have seen an equation with a scattered plot, as the number of binding site decreases, or number of binding site decreases, so there will be low binding of a ligand, and affinity of ligand as it decreases for a receptor binding. So again there will be less biological activity, or we can say the slope, it decreases for that graph, and we have seen that with an equation that how it decreases. With all these 4 examples we have seen one by one. After that, double reciprocal plot. So graphical representation of Lineweaver-Burke equation of enzyme kinetics. So it is advantageous for an calculation of an Km and Vmax. That is, it creates some difficulties with Michaelis-Menten equation, and this can be calculated with this double reciprocal plot easily with accurate information and precise data. After that, we have seen drug receptor interaction forces, and certain drug receptor interaction forces binding with an covalent bonding, ionic or electrostatic interaction. The ion or ion dipole induced interaction. After that, hydrogen bonding. After that, charge transfer interactions. After that, hydrophobic interactions. After that, halogen bonding. After that, van der Waals interactions. So all this drug receptor interaction we have seen. So this was of a summary for measurement of drug response relationship, how a drug response it has been measured. So thank you, my dear students. So we have completed module number 5 and lecture number 8.